Hi, everyone. Welcome to the 113th Stanford MedAI Group Exchange Sessions. This week, we have Kyle Swanson from Stanford University here with us to talk about his work on small molecule drug discovery. So Kyle is a third-year PhD student in computer science at Stanford University, and he's advised by Professor James Zhao. His research focuses on developing AI methods for drug discovery and biomedicine, with a particular emphasis on bridging the gap between computational methods and lab validation. Previously at MIT, where he did his bachelor's and master's, he worked with Professor Regina Barzilay to develop Chemprop, a graph neural network for molecular property prediction, which actually enabled the discovery of Halicin, one of the first antibiotic candidates identified by AI. I'm sure he'll talk a little bit more about that. So Kyle, thank you very much for joining us today. And before we start, actually, do you have any preferences on how you'd like to take questions? Is it okay if we interrupt you, or would you prefer them all at the end?
Yeah, no, please feel free to interrupt with questions at any time.
Awesome. Okay, so I guess let's make this session as interactive as possible. And without further ado, let me hand it over to Kyle.
All right, yeah, thank you so much for having me. Let me just share my screen and then I'll get started. All right, can you see the slides all right? Cool, awesome. All right, yeah, so today I'll be talking about some of our recent work, which is generative AI for designing and validating easily synthesizable and structurally novel antibiotics. So this is work with my advisor, Professor James Zhao, as well as some people at McMaster University as well. So first, as a little bit of context, just on antibiotic discovery. So you might be familiar with this, but the field of antibiotic discovery really started around the 1920s with Alexander Fleming, who kind of accidentally discovered penicillin, which is one of these first antibiotics. He was growing some bacteria in a petri dish, and in this petri dish, it kind of accidentally got infected by this mold colony. And then it turned out that that colony of mold actually excreted this small molecule that ended up killing a lot of the bacteria in that dish. So after Fleming did some more investigation, he identified that particular small molecule and then was able to turn that later on into an effective antibiotic for humans. So that kind of initially started this wave of antibiotic discovery. And in the 1940s to 1960s or so, there were many new classes of antibiotics that were discovered. And you can see some of them on this timeline. So that was very exciting. But after those first few decades of discovery, the field really started to slow down. All the easy to identify antibiotics had already been found, and then it became harder and harder to find really structurally novel antibiotics that would work in new ways and potentially kill different bacterial species. That might not necessarily be a problem because we already have a lot of existing antibiotics, but one of the main issues is actually the rise of antibiotic resistance. So even though we might have some drugs to treat to treat bacteria, over time, the bacteria develop resistance to those drugs just by being exposed and then evolving resistance mechanisms. So there was a study that showed that in 2019, potentially as many as 1.27 million people died specifically because of antibiotic resistance. And the problem is only getting worse. That same study estimated that by 2050, potentially as many as 10 million people might be dying every year because of resistance. And you can kind of see that illustrated in this little timeline on the right. On the left side of the timeline, you can see the year that each of these different antibiotics were introduced into the clinic. And you can see very often within a few years, resistance has started to emerge. That, of course, doesn't mean that that antibiotic is totally useless. There are still many cases where the bacteria will be susceptible to that antibiotic. But if you're infected by one of these strains that does become resistant, that means there might not actually be a drug that could help you. And so that's really a huge problem, of course. So what we're interested in is trying to use machine learning methods to help accelerate the discovery of new antibiotics and specifically structurally novel antibiotics so that that new antibiotic will work in such a way that it can overcome these other existing resistance mechanisms. The first way that you might think about going back about this is an approach that we took a few years back. So in this case, we're using a type of machine learning model that we would call a property prediction model, where it's going to look at small molecules and then try to evaluate their properties, where that property could, for instance, include antibiotic efficacy. So in this particular study, the way we went about it is that we first trained a graph neural network, or a GNN, on about 2,500 molecules that our collaborators had tested in the wet lab. And they had tested each of these molecules to see if each molecule could inhibit the growth of E. coli, a bacterial species. And so among that training set, about 5% of them, I think 120 or so, did actually kill the bacteria. The rest did not kill the bacteria. So we can treat this as a simple binary classification problem where the input is the structure of the small molecule, and then the output is a 1 or a 0, yes or no, did the molecule kill the bacteria? So once we've trained that model, we can then now try to use that to identify new potential antibiotics. And so in this approach, the way we went about it was we took that trained graph neural network, and then we applied it to a library of 6,000 new molecules and basically predicted for each molecule the likelihood that that molecule would inhibit the growth of E. coli. We could then take that list of 6,000 molecules, rank order it from most likely to kill all the way to least likely to kill. And then we took roughly the top 99 predicted molecules and actually tested those in the wet lab to see if those would be effective against E. coli. And what was really exciting for us was that among those top 99 molecules, 50.1% of them actually were active and did kill E. coli. And that's really nice because if you compare that, for instance, to these random molecules, only 5% of those were active. Whereas if you take the ones that are model prioritized, now we're going from 5% all the way to 50% active. So it really helps enrich for these potentially antibiotic molecules. In addition, among those 99 molecules, we found one that was especially interesting. It was this molecule that we later called Halicin. And what was cool about it, this was somewhat unintentional, but a really nice find, was that it didn't just kill E. coli, which is what we trained it to do. It was also capable of killing multiple different bacterial strains, including strains that were multi-drug resistant. And I think part of the reason that that was the case is that when we selected these 99 compounds, we didn't just look for the ones that were most likely to kill. We also tried to find structurally novel molecules, and that made them more likely to kill the bacteria in a new way, and basically a way that the bacteria were not prepared to defend against. So it allowed us to overcome that drug resistance.
Can I ask a quick question?
Yeah, please. Mm-hmm.
So when you created the graph from, like when you're representing a structure of a molecule in a graph, do you also give properties of like the elements and maybe the bond and all of that, or is that somehow not taken in by the model?
Yeah, that's a great question. Yeah, so in terms of the way that the graph neural network works is, I guess if you take this molecule, for instance, as a graph, you can see my mouse, right? Where I'm pointing. Cool. Yeah, so if you take this molecule, for instance, we would create the graph by creating a node for each atom and then an edge for each bond. And to start the graph neural network, we would actually specifically hard code in some properties of the atoms and the bonds. So we compute, like, again, very basic things, but things like the type of the atom, is it nitrogen, hydrogen, sulfur, et cetera. And then very basic things about each of the bonds, like is it single bond, double bond, et cetera. So have a couple like very basic properties of each atom and each bond. And then after that, it's kind of entirely up to the graph neural network to figure out both how to process that information about each atom and bond, and also how to aggregate it across the molecule. So over the course of the graph neural network steps, it'll, for instance, initially look at just this nitrogen, then look at that nitrogen plus its neighboring atoms, then its atoms two bonds away, et cetera, and then build up this overall representation for the whole molecule. So yeah, some very basic like feature engineering as input, but then after that, the model can do what it wants.
Very cool. So I guess even with just the basic information, like you're not giving, I don't know, quantum chemistry details in these.
Yeah, no, nothing like that. Yeah, nothing that requires like fancy calculation, like very, very basic things like the type of atom. Yeah.
Very cool. Thanks.
Yeah, no problem. Looks like there's a question in the chat as well. So how did you establish a molecule structure as novel? That's a good question. So I'll actually come to that a bit later as well, but just as a quick answer, there are a few different ways to, I guess, decide whether a structure is novel compared to existing molecular structures. The way we tend to do it is with this metric called the Tanimoto similarity. So what it does is it will look at two molecules. Again, like imagine this molecule, for instance, and the Tanimoto similarity, it'll first build a kind of a fingerprint, a binary vector that says whether given molecular substructures are in that molecule or not. So for instance, you might have that fingerprint that says this molecule has this particular functional group, it has this ring, has this constellation of atoms, and then it doesn't have a whole bunch of other functional groups. So you get that kind of binary vector. You get the same thing for another molecule. And then you basically see what the overlap is. So to what degree do these two molecules share the same types of substructures? So if another molecule, for instance, also has this ring with like 2 nitrogens, 2 carbons, and a sulfur, then those two molecules be similar. If the other molecule doesn't have that, they'd be more different. And so that'll give you basically a number between 0 and 1, where 1 is they're identical, 0 is they're different. And then you can just set thresholds to say if you're less than 0.5 similar, then we'll consider that structurally novel. So that's generally how we went about that approach. So we'd set a threshold and we say these molecules, according to this 10-molar similarity metric, are relatively different. They mostly do not share the same kinds of structures. Yeah, any other questions at this point? Cool, okay, great. Yeah, so at this point, this was kind of a demonstration of how you can use the property prediction approach. You can train on your training set and then make predictions on a new set of molecules and rapidly identify some potentially exciting new drugs. But we wanted to demonstrate that you can actually go even beyond what someone could do in the wet lab. You could imagine someone could take 6,000 molecules, put that through a high throughput machine and actually test all 6,000, but it'd be much harder to do at a larger scale. So to kind of demonstrate this, what we did was we took our graph neural network and we made predictions on 107 million molecules, which would be extremely hard to actually do in the wet lab. Whereas for us, we could do that in just 4 days of computation on a couple of GPUs. And we went through the same process, made predictions on all 107 million, ranked them by predicted antibiotic efficacy. And then in this case, we tested the top 23 and found 8 structurally novel molecules that seemed to work in the wet lab. So this is very exciting for us. Property prediction does seem like an effective method. But after doing this work, we realized there was still a pretty major limitation. Even though we're able to scale to these large spaces like 100 million molecules, we're not really doing an intelligent search. We're just going one by one through hundreds of millions of molecules, making predictions. And we don't really gain any information based on the molecules we've already predicted. And so you can imagine, even though 100 million is a large chemical space, if you think about the entire space of drug-like molecules, it's much, much bigger than that. People often estimate it's 10 to the 60th molecules. And so there's no way that we can make predictions one by one. So this kind of property prediction approach is not going to scale very well to these much larger chemical spaces. And so we might need a different type of machine learning to handle these larger chemical spaces. So for that reason, we, like many others, have gotten interested more in generative models rather than these property prediction approaches. So I'll just touch very briefly on one example of this approach from my old lab a few years back. It's this junction tree variational autoencoder approach. I won't go into a lot of the details, but the general idea of these generative models in particular is that they can directly generate new molecules. The machine learning itself will design the molecule, and ideally it'll immediately design molecules with the property you're interested in. So you don't have to go one by one through a bunch of bad molecules. You'll just ask the model to give you new molecules, and it'll give you new molecules that have that desired property, which for us could be antibiotic efficacy. And as a quick example of how this might work in this junction tree variational autoencoder approach, the way you would train the model is you would show it a bunch of examples of molecules that have that property. So maybe you showed a bunch of antibiotics. You'll give the model as input the graph of that molecule. In this case, they'll do some like simplification into this junction tree. I'll kind of skip those details. But the point is you can first feed it through an encoder model that will take the molecule and build a neural representation of that molecule. And then you'll have a decoder model that'll go back from that neural representation back to a molecular graph. You'll train it in this kind of reconstruction format of give it a molecular graph, encode it, try to decode back to the same molecular graph. But then when you want to generate new molecules, instead of doing this encoding process, you can kind of sample a random embedding and then ask the model to decode that random embedding. And ideally what it'll do is it'll decode that embedding into a new molecular structure, but one that's still in the same general chemical space as the training set. And so that might give you, for instance, a new antibiotic if you train the model on antibiotics.
But randomly sample, how do you ensure that it would be a new molecule?
You're asking how do you ensure that it's going to give you a new molecule rather than one of the existing ones?
Yeah.
Yeah, that's a great question. I mean, there's definitely a good chance that it could give you an existing molecule. I guess the idea is that this embedding space will be large enough and the model will be trained on enough variety in terms of like molecules as input that it'll be able to still generalize a little bit. But that's a good point. I mean, for instance, if you were to overtrain it, then it might just start to entirely memorize the existing molecular structures. So I think this approach, yeah, you do have to balance that a little bit.
Because I think there is only a very few, like line, like a very fine line between memorization and having a larger data set. Because it would imagine like basically you are subsampling a space that is already learned. There's a kind of like heavy chance that probably the model will probably just sample some existing molecule or existing structure of the molecule.
Yeah, no, I think that is definitely a problem. And I think there are also like some other problems with this kind of approach as well. Like sometimes you also might not necessarily generate valid molecules or something. Yeah, I'll talk about in a second. You also might not get easy to synthesize molecules. So yeah, this has been an approach in the field, but you're totally right. There are a lot of potential limitations to this particular way of doing molecule generation.
Okay.
Yeah, no, great question.
Just a follow-up on that.
Yeah.
How exactly are you ensuring it's the same property? Is it just basically the property that's common across your training set, or are you doing some conditioning on the latent space here?
Yeah, at least in this particular case, the idea is just that you would train on molecules that exclusively have that property. So I guess you're teaching it only to create those kind of molecules. But yeah, there's a large literature, and I think some other people have done more conditional generation where you could like tell it to generate with a given property or a given amount of a property. So yeah, lots of varieties in terms of how exactly you would do this.
Thanks.
Yeah, good question. But what I would say generally, though, is that with this particular approach, as well as many of the other molecule generation methods in the literature, a kind of common limitation across all of them is that they generally create molecules that are actually really difficult to synthesize. It's almost as though these models are too powerful. They're allowed to create almost any arbitrary molecule, and they often don't actually think about how would a chemist go and make that in the wet lab. And so, of course, if you're generating these molecules that are really hard to synthesize, then you can never actually check if they work. And so, in a certain sense, they're almost practically useless. Of course, if you had a lot of time, a lot of resources, maybe you could find someone who'd be willing to synthesize it. But even then, maybe you could only make one or two of them. And it'd be really hard to validate that a lot of the generated molecules have that property that you're interested in. And so we kind of saw this limitation in a lot of the work in the literature, and that really inspired our work to try to overcome this particular problem of generative models that are not creating easily synthesizable molecules. And so that, as I was saying, inspired what we wanted to do, which is to build what you might call synthesis-aware generative models. And so in this task, we have two real goals. So the first thing we want to do is more on the modeling side. So we want to design a generative model for molecules that guarantees that whatever molecules it produces should generally be easy to synthesize. So the first task is the machine learning task of how do you design this particular kind of model. The second question would be, once we have that model in place, what is the workflow that we'd actually use to try to apply this model to a particular drug discovery task? So how would we go about generating, synthesizing, and experimentally validating generated molecules for us for antibiotic design? So I'll walk through each of these steps. First, how we built the model, and then how we actually went about and applied the model. And the core idea of our entire model is really that, again, since we're going for easy synthesizability, we want to take an approach that guarantees that. And so what we'll try to do is have the model specifically design molecules with off-the-shelf molecular building blocks and easy to run chemical reactions. So I'm illustrating that right here. So for instance, maybe you could imagine that your favorite chemical company has these two building blocks, this blue building block and this orange building block. So they already have that on the shelf. They don't even need to make it. It's already ready to go. And maybe they're also very familiar with this particular chemical reaction, which is capable of combining those two building blocks. And so if you have all of this in-house, then you can simply combine those two building blocks with that reaction. You can see the reaction centers in red, and then that'll give you a larger molecule. And so what we're going to try to do is build our generative machine learning model around this idea, where the machine learning model is also going to pick building blocks and select reactions and then build molecules in that fashion, so that you could then go to a chemical company and have them follow that exact procedure to synthesize one of those molecules. And then again, once we have that approach in place, then we can apply it. And for us, what we're going to do is try to apply that to design structurally novel antibiotics for this one particular bacteria called Acinetobacter baumannii. It's a really nasty pathogen. There are few existing treatments. It's very capable of resistance. So we really do need antibiotics for it. So this is our overall approach. And then I'll give a quick overview of a little bit of more of the details. So I'll go into a lot more detail in the next few slides, but this will just give you a sense of overall, what does our generative process look like? So we built a generative model that we're calling SyntheMol. And as SyntheMol really needs a few things as input. So the first thing that it needs is actually a property prediction model. So even though we're doing generative AI, we need an underlying property predictor to help us evaluate the quality of the generated molecules. And so the very first thing we'll do is actually build a property predictor model. So we first go to our wet lab collaborators, ask them to generate a training set for us where they test molecules against bacteria. And then once they have that training set, then we'll train one of our property predictors to predict that same sort of binary classification problem of, does a molecule kill a bacteria, yes or no? So that's one input to our generative model. And then the other thing that our generative model needs access to is a specific chemical space where synthesis is really easy. So again, in a couple of slides, I'll go through all the details. But the idea here is that we want a chemical space where we have a collection of these small molecular building blocks that are on the shelf. We also want to have some easy to run chemical reactions. And then when you combine those things together, that'll give you an actually really large chemical space of potential molecules that could be synthesized. So those two things, the property predictor and the chemical space, they act as input to our generative model, SyntheMol. And then SyntheMol itself, its core challenge is to explore this chemical space in a really intelligent manner so that it doesn't have to go one by one through, let's say, all 30 billion molecules. Instead, it'll intelligently choose some building blocks and some reactions and quickly identify those molecules in this chemical space that have that property that you're interested in. And the specific method we use is a Monte Carlo tree search, and I'll talk through all those details in a couple minutes.
Um,
And
I
yeah,
had a quick question here. So
Mhm.
is it the case that a molecule can be built only by some reaction? Is it basically like a 1-1 mapping, or is it can there be like multiple reactions that give to the same molecule and vice versa? And so I'm curious how that landscape is.
Yeah, it's a good question. I don't think it's necessarily a one-to-one mapping. I'd have to think of some examples, but I could easily imagine that you might have cases where you could have one molecule and it could be made in a few different ways, maybe like two building blocks plus reaction A give you that molecule, maybe a different set of two building blocks plus reaction B give you that molecule. So yeah, I think it's not necessarily a one-to-one. We tend to kind of think of it as a tree structure, which is kind of illustrated here, and it'll become more clear in a few slides of like you pick the first building block, second building block, a reaction that gives you a molecule. But I guess to your question, not all the leaves of that tree will necessarily be unique. You could get to that same molecule a few different ways.
I see. I guess I'm thinking about like if some molecules have a higher probability of appearing if there are multiple reactions that can synthesize them. So will they just be more frequently like generated?
That's a good question. Yeah, I guess we didn't think too carefully about that particular problem because I would guess at most maybe you'll have like 2 or 3 ways of making it. So it'd be like slightly more likely, but still out of the 30 billion, it's not super common.
Okay.
Yeah, so I don't think it'll be a major issue in this case, but yeah, definitely something worth thinking about.
Thank you.
Cool. Yeah, so that'll be our generative model. And then once we've generated some molecules, we'll run this for 20,000 iterations of our generative algorithm to give us around 20,000 or so molecules. Then we can actually go and try to synthesize a few of these molecules and test them in the wet lab. So any other questions about this overview before I go into each of these different steps? Yeah, good. Good question. Okay, if not, I can keep going, but feel free to interrupt anytime. Cool. Yeah, so now we can go through each of these different steps. So first, as I was saying, we want to train a property predictor model. And in order to train the model, of course, we need a training set. So in this case, as I said, our target is going to be Acinetobacter baumannii. So we had our fantastic collaborators go and test a bunch of molecules for us. They took 3 chemical libraries, 2 were drug repurposing libraries, and 1 was a library of synthetic compounds. So they tested each of these against Acinetobacter, about 13,500 molecules in total. Among those, 470 were active against Acinetobacter and prevented the growth of the bacteria. The other 13,000 or so were inactive. You can just see kind of a visualization of the space of these different molecules in different libraries. But in the end, even though it was initially 3 different libraries, we decided to just kind of combine it all together. So it's one single training set with a binary classification task.
Oh, a basic question. What's a library here exactly?
Oh, sorry, library. It's essentially just a collection of molecules. So I think often like various chemical companies will sell these kind of pre-packaged collections of molecules. So for instance, with drug repurposing, it might be, let's say, I think there's one of these libraries that's about like 2,500 molecules. So this company will sell that collection of molecules, and it largely consists of either already FDA approved drugs or maybe some preclinical drugs. So it's drugs that many different people are interested in testing for their own applications.
I see.
Yeah.
They don't share like properties that are similar or something, that they're just owned by a company or released by a company.
Yeah, it's mostly like a company will kind of own this particular collection and sell that as a pack, I guess you could say.
Got it. Okay.
Yeah. Yeah, so these are our training set. And then once we have that training set, we can then train our property predictor models. So we tried actually 3 different types of property predictor models. So the first was a Chemprop model, a graph neural network. So this is the same type of model that I mentioned earlier in our property prediction work. And as I mentioned earlier, the way it works is it takes the molecule's input with the basic features of atoms and bonds, and then it runs a graph neural network on that graph structure, and then predicts the score, which for us will be the likelihood of antibiotic efficacy. Out of curiosity, something we've seen in the past is that you can actually augment these models a little bit to make them a bit more accurate. So what you can do is you can take that graph neural network and then concatenate an additional 200 molecular features computed by this cheminformatics package called RDKit, and that tends to improve performance because now you can have the graph neural network looking at some of the more local structures within that molecule, along with RDKit computing some global features like molecular weight, for instance. And when you combine both of those things with a feed-forward neural network, then you get a little bit more accuracy because the model just has a bit more information. So we were curious to see if that would help improve our property predictor a bit. In addition to that, we were curious to see if a non-neural method would also be competitive. So we tried a random forest model where the input to the random forest model was just those 200 features from RDKit. And then that was fed through the random forest. And interestingly, actually, all the models were pretty similar in terms of accuracy. If you look at the ROC curve or the precision recoil curve, all 3 are kind of essentially pretty close. And I think this might be because all things considered, it's still a relatively small training set, at least compared to a lot of other machine learning where you have maybe millions or billions of inputs. And so at this point, I think the limitation is more on the data side. And so all 3 models are capable of learning something interesting about the molecules. Obviously not perfect, but still doing well enough that they can guide us in the right direction. And I'll return to this point a little bit later once we look at some of the results. But I think one kind of fascinating thing that we discovered is that despite the fact that the performance on the training set is extremely similar, as you can see in these curves, the models, I think, are actually looking at slightly different characteristics of these molecules. Again, maybe the graph neural network is looking a little more locally, RDKit is looking a little more globally. And so what we found is that when we ran our generative model with these 3 different property predictors as kind of the guide for that generative model, it actually led to pretty different looking molecules. But interestingly, again, all of them were kind of equally successful. All 3 methods led us to effective antibiotics, but just structurally different antibiotics. So just kind of like an interesting takeaway that it's not always about getting the most accurate model. Sometimes actually having different architectures is useful just for the purpose of chemical diversity. Yeah, so at this point now we've got our property predictors, so we're ready to go. And then the other thing we need as input for our generative model is the chemical space that we're going to operate within. So we went to this one chemical company that's pretty well known. It's called MEng, and they have this particular chemical space they call the real space, the readily accessible space, that is designed to be very easy to synthesize. And it's also a massive space. So when we did this work, it was 31 billion molecules. I think now it's in the upper 30s or low 40s of billions of molecules. So it's continuing to grow even now. And the way that it's defined is that MEng has 138,000 of those molecular building blocks, and they can combine, let's say, 2 or 3 of them at a time with any one of these 169 chemical reactions. We actually decided that we didn't want to use all of those chemical reactions. We actually had to do some manual work for each reaction, would have taken a little while. And it also turns out that in this 31 billion space of molecules, a vast majority of those molecules can actually be produced with just a few of these chemical reactions. So we found, just looking through the data set, that 13 chemical reactions out of 169 account for 96.6 of those 31 billion molecules. So we decided just to keep things a little bit simple, we'll use just those 13 reactions. We also did a little bit of pre-processing on the building blocks. And so our final chemical space is 132,000 building blocks in those 13 reactions. And that can still give us access to 30 billion easy-to-synthesize molecules. One somewhat challenging thing, though, which we're showing this t-SNE over here, is that if you look at the real space of molecules in black, and even the building blocks in orange, they are somewhat distinct from our training set. Because our training set is more of this like drug repurposing library. And so the chemical characteristics of our training set is a bit different. So we are actually asking our generative model to do a pretty challenging generalization task to go from this training set of chemicals to this somewhat different chemical space in the real space. So it's a bit challenging, maybe leads us to not always get very accurate predictions on all of our generative molecules. But there's at least enough overlap that there is some generalization capability from training set to this chemical space.
What does the training hit refer to?
Sorry, should have explained that. Yeah, so the training set is all 13,000 molecules. The training hits are the subset of 470 molecules in the training set that were active against Acinetobacter. So just giving a sense, like for instance, this is a big cluster of kind of similar molecules that all kill Acinetobacter. So I guess ideally what you'd want to see is a bunch of these red molecules within the black space, because that would tell you that your likely effective molecules are within that same chemical space. But again, a bit of the challenge is there aren't a lot of examples there. So we're going to have to hope that the model can actually generalize very well and understand within this new chemical space what would be active and what would not be active. Cool. Great. So these are now our two inputs. So we have our property prediction model to evaluate molecules, and we now have this large chemical space where we can easily synthesize the molecules. So at that point, now we can get to our generative model. And so you might think the first thing that might come to mind is we have this really large chemical space. We want to design molecules. What would be the easiest thing that we could try to do to get to good molecules? The first thought might be to do a kind of greedy approach where we'll look at our building blocks and maybe we'll just score each building block. We'll ask our property predictor, what do you think of all these individual building blocks? Do you think those will be effective against Acinetobacter? And then we'll simply take the building blocks that have the highest scores. So maybe take the very best building block, second best building block, find a chemical reaction that can combine them, and that'll be our molecule. And we'll hope that that'll be really effective. So that could potentially work, but we found that there were a few problems with that particular approach. The first thing is that when you look on the building block level, there are actually very few building blocks that have high predicted antibiotic efficacy. That's what we're showing over here in this chart right here is all the 132,000 building blocks and their predicted antibiotic efficacy. You can see only a small fraction of them have a chance, according to our model, of killing Acinetobacter. So if all we did was just choose the high scoring building blocks, we'd have very little chemical diversity. Whereas we really want the best chance of finding something good, so we don't want to limit ourselves to just one very small part of chemical space. So that's one potential limitation. And then the other limitation we saw is that even if your building block has strong potential for antibiotic efficacy, that doesn't necessarily mean that the full molecule that contains that building block will also have antibiotic efficacy. And so that's what we're trying to show over here, where on the x-axis you can show, or maybe start with the y-axis. On the y-axis, we'll take a molecule from the real space, one of those 30 billion molecules. We'll ask our property predictor, what do you think this molecule's probability of killing Acinetobacter is? So that's stat value. And then on the x-axis, we'll decompose that molecule into its constituent building blocks, let's say 2 or 3 building blocks. And we'll ask, on average, what is the likelihood that those building blocks will kill the bacteria? And you can see there's a very bad, basically no correlation between those two things. So even if you select, let's say, a couple really good building blocks, that could give you a terrible full molecule. Or you could select a couple really bad building blocks, and that'll give you a really great full molecule. And that's because once you combine these building blocks, I mean, obviously the shape of that full molecule will be different. Some of its chemical properties will be a bit different. And so there's not necessarily, you can't necessarily get to good molecules just by selecting good building blocks. It will maybe potentially get you in the right direction, but that's not sufficient. So for those reasons, we believe that this greedy approach will not make a lot of sense. And so instead, we wanted to use a smarter algorithm for trying to generate these molecules. And so the method that we went with is this well-studied method called the Monte Carlo tree search. or MCTS. And what's really nice about this method is that it kind of has two benefits that counter these two problems. The first is that as part of the algorithm, it necessarily does a lot of exploration, where it will try to construct very diverse molecules in order to explore the space and get a sense of what molecules are good and what are bad. So that'll automatically give us some diversity in the molecules that are generated. But on top of that, it doesn't just do exploration, it obviously also tries to do exploitation, where it'll try to look at the full molecules that it has generated so far. It'll evaluate them and then try to use information about those full molecules to further guide its search. So when it finds good molecules, ones that are likely to kill Acinetobacter, it'll then try to generate similar molecules. When it finds some really bad molecules, it knows to maybe avoid that part of chemical space and move in a new direction. And so that'll ideally try to get us to parts of chemical space where we have these great molecules. So that's the algorithm that we used. And then in terms of how this actually looks in terms of generation, this is kind of how our SyntheMol model works. So it's going to be a Monte Carlo tree search guided by our trained property predictor. So here I'll walk you through how one iteration of this algorithm would work. We start up here at the root of this tree that we're searching. So at this point, we kind of haven't done anything. This is our starting point. Then the very first thing we have to do is we have to decide what is the first building block that we want to add to our molecule. So on level 1, we'll list out all 132,000 building blocks. Here I'm just showing 3 of them. And we'll score them according to this Monte Carlo tree search score. So I'll explain the score in the end, but for now, I'd assume that there is a way to score each building block. So we'll score each of these building blocks, and then we'll figure out which one has the highest score, in this case, this blue building block, and we'll select that as our first building block. Once we've done that, now we can figure out what is the next building block that we want. We'll take this first one, and we'll couple it with every other one of these 132,000 building blocks. So we this building block plus itself, this building block plus the second one, this plus the third one, and so on and so forth. And now what we're trying to do is we're trying to score combinations of building blocks to figure out which combination will be the best. In this case, once you're on this second level, there will potentially be some combinations where those two building blocks simply cannot be combined. We don't have a chemical reaction in our set that's capable of combining them, so we'll just kind of throw those away and ignore them. Among the combinations that are feasible, where there is some way to combine those two building blocks, we'll then again score them with the scoring function that I'll explain in a second, select the one with the highest score, and then proceed from there. Sometimes we'll add yet another building block, a third building block, but here I'm illustrating a case of just 2 building blocks. And once we've selected those 2, now we can apply a particular chemical reaction that is compatible with those building blocks to give you a single molecule that contains both of those building blocks. And now that we've gotten to this full molecule, we can run that through our property predictor, and it'll tell us how good that molecule is. In this case, it's a pretty good one, it has a 0.9 chance of inhibiting the growth of Acinetobacter. And that will actually then provide feedback to the rest of the Monte Carlo tree search to tell it that this was a good molecule and maybe it should select those building blocks again. So this score will feed back into the full molecule as well as each of the individual building block selection components. And it'll feed back and basically say, these are good, we should do that again. So that's how the workflow would work, assuming you have this Monte Carlo tree search scoring function. And so now I'll explain what that specific scoring function is. So I'm drawing this equation right here. So the way we score a particular node n, so like one of these blocks right here, is we'll look at 4 components of that node. The first thing we'll look at is the exploit score. So this is asking for this node, for this building block, what is the average score of full molecules that are produced by this node? So for instance, with this building block here, for this particular case, it found this full molecule that has a score of 0.9, but maybe in some other cases, it maybe found a score of 0.5, 0.3. And so this exploit score is going to be that average. So it's basically the potential of that building block to give you good or bad molecules. In addition to that, we're also going to look at the individual property prediction score of that building block itself. As I said before, that's not necessarily super well correlated with the final molecule score, but we find that this is actually still pretty helpful. So in that case, we'll score this particular building block and see its particular antibiotic efficacy potential. On top of that, we also have an explore function. So this will basically try to encourage the model to select building blocks that it hasn't seen before. So we're going to keep track of throughout this algorithm as we go through multiple iterations, how often have we seen each of these building blocks compared to each of its neighbors. So in this case, for instance, this building block had already been selected twice before in previous iterations, this one only once before, this one initially once, now we just selected again. And so we're going to try to preferentially select building blocks that have not been seen too often in order to collect more information about whether they're good or bad building blocks. I think these three things are somewhat standard in the literature, but then one thing that we added on top of this is this extra diversity penalty. It kind of helps alongside this explorer function. And we tried to really hard code and make sure that the algorithm doesn't use the same building blocks too often. So we keep track of, regardless where in the selection process this happens, each building block that is selected. And we keep track of how often every building block was selected. And if a building block is chosen too many times, like if it gets past, let's say, 20 or 30 times, then we start to really penalize selecting that building block again so that we don't get a lot of the same molecule. To be honest, this was more of an empirical discovery. We initially had these 3 criterion, and then we found that the model just loved like 1 or 2 building blocks, selected those again and again and again. And it's very possible those are super effective. But for our purposes, we would really be interested in having a variety of building blocks, a variety of molecules. So we added this additional penalty to get us to extra diversity. So our scoring function throughout this search is going to be a combination of those factors. So any questions about how the Monte Carlo tree search approach works?
Quick question on the computational complexity of this. Like, are you already, so I know that to do the exploit, you kind of need to go to the depth, but do you actually do that, or do you have like a heuristic to kind of simulate that?
Yeah, good question. So the exploit is, I guess, a score that is like updated as you run the algorithm. So it's kind of simultaneously as you're generating, you're also collecting information. So yeah, on the very first iteration, you actually have no information. Every single building block has an exploit score of 0. And like we don't have any initial heuristics, but that's actually kind of why we used a property score, because the property score is something you can pre-compute about every building block, gives you that vague sense of what is maybe slightly a good building block, what's a bad one. So I think this helps, especially early on in the algorithm, to point you in the right direction. And then, yeah, over time, as you do generation, the exploit score gains more information, gets more accurate, and then starts to be more useful in terms of guiding you in the right direction.
Got it. Okay, thanks.
One quick question. Do you have a upper limit on how many building blocks you combine? Like, do you know from biology that, okay, maybe I don't need more than 10 building blocks or 15 building blocks?
Yeah, that's a great question. So we have 2 answers. I guess the first answer is maybe the biological answer, which is if you look at our training set of molecules, I guess those molecules are not directly constructed by these building blocks. Those are like other random FDA molecules, could have been synthesized in many other ways. But in terms of the size of those molecules, that corresponds to anywhere between 2 and 6 building blocks. So biologically, you'd expect roughly that size. Any larger, and then it might be really hard to get the molecule across the bacterial membrane into the cell. But then there's the second answer, which is the kind of logistical chemical synthesis answer, which is with this company and with other companies that do a similar type of chemical synthesis, they have hyper optimized their ability to combine 2 or 3 building blocks with 1 chemical reaction. Anything more than that, and suddenly your costs go up a lot, the time to synthesize goes up a lot. And so we've actually decided to limit our algorithm to just 2 or 3 building blocks and 1 chemical reaction. It still gets you through those 30 billion molecules, but that's what we've decided. So yeah, I guess algorithmically speaking, and also in terms of the actual code that we wrote, there is no limit, but we kind of put in at that limit of one reaction and a few building blocks in order to just make it like feasible to synthesize.
Thank you.
Yeah, great question there. Cool. Yeah, so if there are no other questions, this is our overall SyntheMol method. And now that we have this method in place, now we can actually use it to try to design new antibiotics. So what we did was we took SyntheMol and we ran it for 20,000 rollouts, so 20,000 iterations of that algorithm I just described. And we actually did that separately for each of those 3 property predictors, for the Chemprop party kit, and the random forest. So it's sort of like 3 independent experiments, each one for 20,000 rollouts. And then in each of these cases, we now have a collection of a little over 20,000 molecules for each property predictor. And that's actually way too many to synthesize. It'd be super expensive, would take a long time. And also not every generative molecule is going to be a great one, because of course the Monte Carlo tree search has to do a lot of exploration. And when it explores, it'll make a bunch of mistakes along the way. So what we want to do now is go from that 20,000 something molecules down to a set of, let's say, around 50 or so molecules that we are actually interested in synthesizing and testing. So to do that, we put 3 filters in place to select those optimal molecules. The first filter is on novelty. So we really want to avoid simply generating analogs of known antibiotics, because if we generate an analog, then as soon as the bacteria is resistant to that initial antibiotic, they'll probably be resistant to our molecule as well. So this ties into that similarity question. So the way we do that here is we use this metric called diversity similarity. It'll measure basically the structural similarity between our generated molecule and each of the known antibiotics in our training set. And we have a hard threshold of 0.5. So we're not going to allow our generated molecule to be more than 0.5 similar to any existing antibiotics. Once we've done that novelty filtering, we can then do filtering on predicted efficacy. We can look at the property prediction scores on each of our generated molecules, and we'll only keep the generated molecules that are most likely to be effective against the bacteria. The actual threshold here was a bit of a heuristic. It just felt to us that maybe if we looked at the top 20% of molecules by predicted antibiotic efficacy, that would get us in a good range. And you could certainly tweak this depending on how much you want to prioritize effectiveness here. And then once we've done that, we now have novel and predicted effective molecules. And now we just want to select a diverse set of those molecules, because here we want to avoid analogs of the same compound, because that's not super interesting. So what we did here was, among those novel effective molecules, we did a k-means clustering using tandem motor similarity as the distance metric between molecules. And we used k equals 50. So it gave us 50 clusters of molecules. And within each cluster, we simply selected the highest scoring molecule from that cluster. So it should be 50 novel molecules that are all diverse and should all be ideally effective against the bacteria. And so we did this for each of those 3 property predictors. So for each one, we got 50 molecules, so it's 150 molecules in total. And just to show some of the metrics on these molecules, this is looking at just the results from one of the property predictors, so SyntheMol plus the Chemprop property predictor as its guide. And what I'm showing is the initial generated set and then the final selected set after going through the filters. So here, this is focusing on novelty. So we're looking at the similarity between the generated molecules and the active molecules from our training set, the hit molecules. And you can see initially, sometimes we have some somewhat similar ones, but obviously after implementing this hard threshold, now we only have the more dissimilar ones, the more novel molecules. On the effectiveness side, this is actually kind of an interesting point. If you look at our initial generated set of molecules, you can see we do get a nice range of scores, including some higher scores, so more likely to be antibiotic effective. When we focus in on just the top 20% of scores, you can see now we're really emphasizing the high scoring molecules. And as a point of comparison, what we did was we sampled 25,000 molecules at random from that real space of 30 billion molecules and scored those with our antibiotic predictor. And so you can see if you take randomly selected molecules, a vast majority of them have almost no chance of being effective antibiotics. Whereas if you look at our generated molecules, they're much more likely to be effective. So this is already an indication, at least in silico, that our model is really guiding us towards the part of chemical space where there are more effective molecules. And then the last thing over here is just focusing on the chemical diversity component. So this is saying among the generated molecules or among our final selected filtered list, what is the similarity between pairs of molecules on average? So again, after applying that diversity filter, we tend to get more dissimilar molecules, which is what we're looking for. And this final training t-SNE over here gets to that point that I mentioned earlier, which is we have SyntheMol with these 3 different property predictors. 3 different property predictors are all generally roughly equal in terms of training set accuracy, but they do lead to different types of chemicals when doing generation. So you can see there's somewhat some overlap between the molecules generated by these 3 different predictors, but they are also decently different. So that was kind of cool for us to see that we got extra diversity just by using different property predictors. And so now that we have these molecules, we can actually go ahead and do synthesis. So as I said, we selected 150 molecules total, 50 from each of those 3 models. And in the end, we were able to request 70 of them from enamine. So not all 150 were directly available from enamine. This gets into kind of some chemical details, but essentially the reactions that we were able to hard code into our SyntheMol system were actually a little more broad than the specific reactions that enamine itself is capable of. There are tons of chemical details I can talk about if you're curious, but basically we're not able to directly synthesize all of those molecules. But we were still able to at least ask for 70 of those 150. And it's still much better than pretty much any other generative model out there. Once we got to those 70, so now those are 70 that enamine says it is capable of making, we were then able to successfully synthesize 58 of them, so about 83% success in just 4 weeks. And so that's still relatively rapid, it was pretty cheap, and it's a pretty good success rate among the ones that enamine tried to synthesize. Among those 58, we had 26 that were from SyntheMol with Chemprop, 22 that were SyntheMol plus Chemprop RD kit, and then 10 that were SyntheMol plus random forest. And so now we can finally get to the kind of exciting and scary part a little bit as well, which is experimental validation. So seeing if the molecules actually work or not. So the first test we did was to test those generated molecules against Acinetobacter baumannii. And so this is the exact same growth inhibition assay that we used to create the training set, run by the same people at McMaster University. And so initially, actually, none of the molecules worked out of the box. We tried those 58 that we got synthesized. And here what I'm showing is for each of those 58 molecules, I'm showing the minimum inhibitory concentration. So basically the least amount of molecule that you can deliver and still see some sort of effect against the growth of the bacteria. And here a higher number is a bad thing because that means you need tons and tons of molecule to have any effect on the bacteria. And here we actually totally max out. We give as much molecule as we can and nothing is really working. So first, this is a little concerning to us because it seems like maybe actually nothing's working after all. But luckily, our collaborators had some really good insight into what might be going on. And the challenge is that killing bacteria is actually a very hard thing, in particular when you're going after these gram-negative bacteria, and Acinetobacter is one of these, because you have to do two things, not just one thing. The first thing you have to do is your molecule has to actually get through the cell membrane into the bacterial cell. And that's especially hard in these gram-negatives because they have not one, but two cell walls. They have to go through twice and get through potentially in two different ways in order to get into the inside of the cell. And then once you're on the inside of the cell, then you also have to have some sort of activity against the bacteria. So for instance, you maybe have to inhibit the functioning of some essential component like some essential protein. And so what our collaborators were thinking is that maybe what's going on is that our molecules do have activity. It's just that they can't get inside the cell. And of course, if they can't get inside, then they can't exert their activity and they can't actually kill the bacteria. So they were thinking, what if there's some way that we can maybe help the molecules get inside and then see if they do have this activity or not? And so the experiment that they ran is they then tried to couple each of our generated molecules with a known permeabilizer, where the permeabilizer is another small molecule that can help open up the membrane a little bit. It will not kill the bacteria, but it'll simply open up the membrane and allow our molecules to get in. And so once you do that, then the exciting thing is we actually found that 6 of our 58 molecules are actually very, very potent. So they have a lot of that activity. They were just struggling with the permeability component. So here what we're showing, the row 1 is that initial experiment, no permeabilizer. Rows 2 and 3 are experiments of each of our molecules with one of 2 different permeabilizers, either SPR741 or colistin. And the 6 in bold are the ones where you can see they actually have really exciting, very low minimum inhibitory concentrations. So they're actually quite effective. So this is very exciting for us. But of course, one thing that we have to check is that we didn't just get totally lucky and maybe we just happened to randomly get some good molecules. So the point of comparison, we selected 58 molecules at random from that same chemical space and then tested their activity. And so you can see when you look at the random molecules, some of them have a tiny bit of activity, at least when you include the permeabilizer, but it's nothing nearly as potent as our 6 identified molecules. So this was a really good control for us that it's because our model is selecting for these really effective molecules that we're then finding this high potency in vitro. Out of curiosity, what we then wanted to see, is this just limited to Acinetobacter, or did we potentially find some broad spectrum antibiotics? So we looked at those 6 exciting molecules and then tested them against some other bacterial species. And we found that, in fact, they do have broad spectrum efficacy, again, with the permeabilizer. So just to go through quickly, we tested them against Acinetobacter. We also tried E. coli, Pseudomonas, Klebsiella, and Staph aureus. And so in each of those cases, generally, if you include the permeabilizer, then you do have activity. Pseudomonas is a little more challenging. Only 1 molecule worked there. but at least across some of the other species, we were able to see activity. And then the very last experiment that we did was to look and see if there's any sort of synergy between our permeabilizer and each of our identified molecules. So each of the columns corresponds to one of our 6 molecules, and then each of the rows is one of these species along with the permeabilizer. And what we're looking for here is when you see this sort of L shape, what that is telling you is that you can actually get away with very low doses of both molecules and have a strong effect. So for instance, as you go across the x-axis and up the y-axis, the dose of either our molecule on the x-axis or the permeabilizer on the y-axis is increasing. And you can see even if we have very low doses of each, as long as we have a bit of both, then you actually have pretty strong efficacy. So that was pretty exciting for us, is because if you can get away with low doses, there's a much greater chance that you won't have toxic side effects because you actually don't have to deliver too much of either drug to the person taking the drug. So just as a quick summary of what we've talked about, what we built in this work was SyntheMol, which is the synthesis-aware generative model for drug design. And the key idea is that it uses a trained property predictor along with the Monte Carlo tree search to explore these really vast chemical spaces for exciting new molecules. We also introduced some filters to focus on novel, effective, and diverse generative molecules. And the really exciting part is that we were able to then synthesize and experimentally validate 58 of these generative molecules. Among those 58, we found 6 highly potent and structurally novel antibiotic candidates. So we're really excited about the potential of this model to help with drug discovery. So I'll end there, and I'm happy to answer any questions.
Awesome.
Yeah, I had a quick question.
Yeah, sure.
So if I guess what I'm trying to ask is, for the bacteria, is the I guess being more resistant to drugs, is that more of the permeability issue or the inhibition issue?
Gotcha. Yeah, for the resistance, I think that's more commonly an issue with, I guess, whether the small molecule still has activity or not. I mean, there might be examples where maybe the bacteria, there probably are some examples where the bacteria can evolve to maybe prevent the small molecule from getting in. But I think the more common resistance mechanism is that, let's say you have this small molecule that gets inside the cell, it hits some essential protein and kind of knocks out that protein. If the bacteria can now, let's say, slightly mutate that protein so that the small molecule can no longer bind to the protein, then it'll become resistant to that drug. The drug will still get inside the cell, but it can no longer bind and have its effect. So I think that's the more common mechanism.
Okay. Yeah, because I was thinking it would be interesting to see, because I'm guessing that most of the drug discoveries were for the inhibition side. So it'd be interesting to see if there is, I guess, a drug that could make it more permeable so that even if the currently resistant drugs are less, I guess, effective, you could just essentially douse it with more.
Yeah, no, that's a great question. I think that might be true. I don't have all the knowledge there. I'd have to ask my collaborators because they're much more familiar with the antibiotic space. But I know, for instance, one of the permeabilizers we used, SPR741, I think it's currently undergoing or maybe just finished a phase 2 trial to see if it can be used in conjunction with existing antibiotics to make them more effective. So it sounds like that is potentially one approach, is just add this permeabilizer to make existing drugs more effective.
Yeah, because I mean, even on the previous couple slides, I think there was the random ones with the permeability. I think some of the random ones still had some effectiveness. So
yeah, that's true. Yeah, I mean, definitely not like super high effectiveness, but yeah, the permeabilizer certainly does help a little bit. Yeah.
Right. Okay. Thank you.
Cool. Thanks.
Yeah.
I see there's also a question in the chat as well. I think you're asking, could knowledge regarding the need of improved permeability influence model design, perhaps use it in a property predictor? Yeah, no, that's a great question. So we thought about this a lot, especially once we got those initial results where we didn't see any effectiveness. We were then wondering if there's a way to incorporate that permeability more directly into the property predictor. I think the answer would be yes, if you can get the data, basically. It sounds like it's generally hard to get really good permeability data for bacteria in particular. I think the assays that you have to run are just a little challenging to do very precisely. But absolutely, if you could get access to that data, then I think you would certainly train up a model and then add that as an additional objective in your generative model. And just to give an example of that, in some of the follow-up work we're doing, we don't unfortunately have access to great permeability data sets, but we did notice that in this first round, a lot of our molecules were not particularly soluble, which is also pretty important for drugs. So in our new work, what we're trying to do is a bit of a multi-parameter objective of both antibacterial efficacy and also solubility, since we can train some pretty good solubility predictors. So yeah, I think as long as the data is there, then I think that would be a great approach.
Cool. Thanks so much, Kyle. I think this was a wonderful talk. I guess we are a little over time, but if you do have a few minutes, we can leave the floor open for questions. But before that, let's thank our speaker with a round of virtual applause. And yeah, maybe we can take one question or something if anybody has it.
Yeah, sounds great. And thanks again for having me. It was a lot of fun.
You applied this for bacteria. There's a lot of interest to apply small cell medicines for multiple sclerosis, like autoimmune disorders. What would you need to do, know to do a similar experiment, but for autoimmune disease like multiple sclerosis?
Yeah, no, great question. We've also been trying to work on not that particular application, but alternate applications of this model outside of antibiotic efficacy. And I guess the nice thing about these sorts of machine learning approaches is that they are pretty easy to generalize to other types of drug discovery. So really, I mean, if you're looking at this overview slide, essentially the only thing that really needs to change is that initial training set. All the machine learning mechanisms can kind of stay the same. The property predictors we've shown in the past, they're very capable of learning all sorts of molecular properties. The generative model is kind of agnostic to that. Just whatever reward you train it on, it'll learn to do that. Chemical space doesn't really need to be modified. So as long as you had a really good training set for multiple sclerosis, where you had effective molecules and not effective molecules, then you could potentially use all the rest of the mechanisms to try to solve that same problem. Of course, there might be other bottlenecks that come along the way. As I was just talking about in the antibiotic case, often there are many different properties you need, like solubility, toxicity, or non-toxicity, et cetera. But again, like as long as you have that training data, then I think a lot of the same machine learning could potentially be applied. And I guess I should have mentioned as well, like all this code is open source as well. So if you have the chance, it'd be great to try that out and see if it works.
Yeah.
Awesome. So I guess if folks have more questions, you can reach out to Kyle or reach out to us, and we'd be happy to put you in touch with him. We'll have the video up on YouTube later today or this week. So feel free to check it out. And then if you come up with more questions, reach out. But thanks again, Kyle, and thanks everyone for joining today. And I'll see you all next week.
Thank you so much, everyone.